Chapter 16 in Molecular Diagnostics covers quality assurance and quality control in the molecular laboratory. Quality assurance and quality control are concepts that are universal throughout the clinical laboratory and are based on standards established by CLIA, the Clinical Laboratory Improvement Amendments, in 1988. CLIA specifies quality standards for proficiency testing, patient test management, quality control, personnel qualifications, and quality assurance for laboratories. In this chapter, we'll review these concepts. The objectives we'll be covering in this chapter are describing proper specimen accessioning for molecular testing, describing the optimal conditions for holding and storing specimens and nucleic acid, explaining the basic components of molecular test performance, including quality assurance and controls, discussing instrument maintenance, repair, and calibration, describing recommendations for preparation and use of reagents in the molecular laboratory, and explaining documentation and reporting of results. Some of the concepts we'll be covering are applicable to all clinical laboratory testing, and some are specific to molecular. Molecular tests, like any clinical laboratory test, require optimal specimen handling and processing for accurate and consistent results. The success of a test procedure is affected by the age, type, and condition of specimens. So specimen collection, transport, and handling in the laboratory require careful attention. The condition of the specimen, and if necessary, the chain of custody, is reviewed upon receipt in the laboratory. No specimens should be accepted without proper labeling and identification. And if a specimen is unacceptable, the disposal or retention of the specimen should be recorded. These factors are considered pre-analytical and can cause pre-analytical error when processing the samples. Pre-analytical error is the consequence of erroneous or misleading results caused by events that occur prior to sample analysis. Precautions need to be taken when handling all clinical laboratory specimens, as they are potentially infectious and should be handled with standard precautions using proper personal protective equipment. Gloves are highly recommended and most of the time required, not only as part of standard precautions, but in regards to molecular testing, they're required to protect nucleic acids from nuclease degradation. Gloves are absolutely required for handling of RNA, as we saw in previous chapters with discussions of RNase-free equipment and reagents. Transmission-based precautions also should be considered, including respirators, when airborne or contact transmissible agents are being handled. Contact precautions are designed for direct patient care and are not necessarily applicable at all times in the laboratory. Molecular testing specimens include sources that we've discussed in previous chapters, such as blood, bone marrow, cerebrospinal fluid, and tissue, and these can be fresh, frozen, or even fixed tissue in some cases. Molecular amplification methods have enabled laboratory professionals to perform nucleic acid testing on specimens with minimal cellular content, such as cerebrospinal fluids. These samples are centrifuged to collect the cells before DNA or RNA is extracted. Cross-contamination of molecular specimens should be avoided. It can most likely occur from pipetting carryover. Also, once aliquots are removed from a specimen, they should never be returned to the original tube or vessel. Molecular specimens should also be inspected for hemolysis. Hemoglobin inhibits enzyme activity, so hemoglobin and anticoagulants should be removed effectively in most DNA and RNA isolation procedures. If white blood cell lysis has occurred, the DNA or RNA yields may be reduced, and this could result in false negative results in qualitative testing or inaccurate measurements in quantitative analysis. Solid tissue samples are best analyzed from fresh or frozen tissues, although, as mentioned before, fixed tissues can be used, but the quality of nucleic acid depends on the fixing process and the fixative used. Phlebotomy collection tubes are available with a number of different additives designed for various types of clinical tests. Table 16.1 in your text lists a selection of collection tubes commonly used for molecular biology studies. Some anticoagulants used in blood and bone marrow collection may adversely affect analytical results. Heparin and occasionally acid citrose dextrose (ACD) tubes have been shown to inhibit enzymes used in molecular analysis, such as reverse transcriptases and DNA polymerases in vitro. Due to the possible effects of heparin, trisodium EDTA, the lavender top tube, or acid citrate dextrose, the yellow top tubes, are recommended for most nucleic acid assays involving enzymatic treatment of sample nucleic acid. The general rules for holding specimens for processing differ depending on the analyte and its stability in the cell. Many molecular methods do not require integrity of tissue or cell structure, only that the nucleic acids remain intact. Table 16.2 outlines specimen storage requirements for nucleic acid extraction and is summarized on the next few slides. Blood, bone marrow, and fluid samples can be held for less than a day at 22 to 25 degrees Celsius. So these samples are stable at room temperature. They can be held longer for 3 days at 4 degrees Celsius refrigerated. When the buffy coat is extracted and centrifuged down from these samples, they're stable. It can be held for 1 year at freezer minus 20 degrees or greater than a year at minus 70 degrees. It's not recommended that tissue samples be stored at room temperature, and they're stable for less than a day refrigerated at 4 degrees, stable for greater than 2 weeks at minus 20 degrees, and stable for greater than 2 years at minus 70 degrees. Isolated DNA samples are stable at refrigeration to room temperature within 26 weeks, and they're stable for 1 to 3 years at refrigeration temperatures of 4 degrees, and less than 7 years at freezer temperatures of minus 20 and minus 70 degrees. These specimen holding and storage requirements are specifically for DNA samples. Specimen holding and storage for RNA samples vary from DNA. With both DNA and RNA, the long-term survival of the sample depends on the initial isolation and handling of the specimen. So for samples that will be processed for RNA testing, such as the blood, bone marrow, and fluid samples, they're stable for less than 2 hours at 23 degrees room temperature or 4 degrees refrigeration. If these samples are placed in denaturant, they're stable for 5 days at room temperature and 7 days refrigerated. They can also be stable for 1 to 2 weeks in denaturant at minus 70 degrees. Tissue samples for RNA testing are stable for less than 2 hours at 4 degrees refrigeration. They can be stable for up to 2 years frozen at minus 70. Isolated RNA samples can be stable for less than 30 days at minus 20 and minus 70 degrees, and greater than 6 months at minus 70 degrees in ethanol. It's not recommended to store isolated DNA at refrigeration or room temperature. We've seen this slide before regarding preparation for RNA analysis, and it was mentioned previously that it's important that bench and equipment need to be designated RNase-free or RNF. Wipes with RNase ZAP or RNase away products can be used to ensure RNase-free equipment and environments. Disposables also need to be certified as RNase free and can be rinsed with 1% diethyl pyrocarbonate. Reagents as well should be certified as RNase free and can be tested with RNase alert to ensure that they are RNase free. So up to this point, we've been discussing pre-analytical factors such as specimen collection and storage. Now we'll talk about actual test performance. So according to the FDA, an approved test is one that has been accepted by the Food and Drug Administration as safe and effective for its intended use based on the manufacturer's data. The manufacturer conducts studies to show that the test performs as claimed and does not present any unreasonable risk. An FDA-cleared test is one that has been shown to be similar to other tests already marketed based on manufacturer's data. Even though the manufacturer conducts studies on their tests, laboratories are required to validate testing once they choose to adopt the test. The FDA requires validation of performance of clinical tests and reagents in accurately detecting or measuring analytes prior to use in human testing. Many tests in the molecular laboratory are independently designed or are adaptations based on published methods. Development of new tests in the clinical laboratory requires validation of the performance of the method and reagents in accurately detecting or measuring the analyte. Test performance is assessed by several criteria. Analytic sensitivity is evaluating the change in an assay response with the corresponding change in an analyte. Analytic specificity is the ability to detect the analyte itself and not non-specific targets. Clinical sensitivity and specificity will be discussed a little bit more on the next slide. Detection limits evaluate the lower limit detection of the analyte. Accuracy is evaluated to ensure that the method can produce correct results, and precision is evaluated to see that the results agree between independent test results. Reproducibility shows that the test results are consistent by the same procedure. The AMR is the range within which a specimen may be measured directly without dilution or concentration. The reportable range is the range within which test results are considered to be valid with or without dilution. The reference range is the expected analyte frequency or levels from a population of individuals. And linearity is the quantitative correlation between test results and actual amounts of the analyte. The clinical sensitivity and specificity of test or method needs to be evaluated as well when validating testing. Clinical sensitivity is the ability of the test result to predict a clinical condition. The formula for calculating clinical sensitivity is true positives over true positives plus false negatives times 100. So for example, if 95 out of 100 patients with a gene mutation have a disease state, the test has a clinical sensitivity of 95%. Clinical specificity is defined as disease-associated results in patients who actually have disease conditions. The formula to calculate specificity is true negatives over true negative plus false positive times 100. An example of this would be 1 of 100 normal specimens displayed a gene mutation, with 1 being a false positive, so there's a clinical specificity of 99%. Accuracy is evaluating that a method produces correct results. Calculating accuracy uses the formula true negatives plus true positive over true negative plus true positive plus false negative plus false positive times 100. So these are formulas and concepts that we also saw in general lab principles, but we're reiterating this information because it's important components of test method validation, and evaluation. When evaluating new tests, methods, or instruments, validation of specimen types that will routinely be encountered must be performed. FDA-approved and cleared tests will specify the validated specimen types, but once a test is brought into a clinical or diagnostic laboratory, the end user must also evaluate the approved specimen types. The number of specimens tested varies depending on the procedure and the availability of the test material. For example, validations of at least 40 samples of venous blood are more readily and easily attainable, opposed to those that require more difficult or less frequently collected samples, such as cerebrospinal fluid, tissue biopsies, or even arterial blood gas samples. When commercially developed and FDA-approved molecular methods are being validated, they're verified by using the purchased reagent sets created by the manufacturer to test validation specimens in the laboratory. If the commercial test is modified, validation is then required to show equal or superior performance of the modified procedure. And once a procedure has been established, the method is documented in the laboratory according to Clinical and Laboratory Standards Institute guidelines in specific policies and procedures. Proficiency testing can be performed as part of validation studies, but should also be routinely performed in the laboratory. Proficiency testing refers to the analysis of external specimens supplied to independent laboratories from a reference source. This testing is done to both assess the skills or competency of laboratory personnel performing molecular assays and also to assess the performance of the assay itself. The College of American Pathologists, or CAP, which is our laboratory accrediting agency, and other organizations supply specimens for molecular analysis for proficiency testing. They supply samples such as those for molecular genetics, molecular oncology, and molecular microbiology testing. Although a number of analytes are not available, especially for those tests that are offered in small numbers of laboratories. So if proficiency specimens are not commercially available, laboratories must establish another proficiency testing procedure. One way that this can be done is by exchanging split specimens between two or more laboratories. Alternatively, specimens measured or documented by independent means, such as chart review, can be tested within a single laboratory. Proficiency testing is also particularly important in molecular testing for those FDA-modified tests. In these situations, proficiency testing assures the accuracy and reproducibility of the test results and that they're satisfactory for patient testing. Controls are another important component of molecular diagnostic testing that are required to be performed as part of validation studies, but also routinely in molecular testing. Controls are samples of known type or amount that are treated like and run with patient samples. Interpretation of test results always includes inspection of controls and standards to verify acceptable test performance. With qualitative tests, a positive, negative, and in some cases a sensitivity control are required. The sensitivity control defines the lower limit of the analytical range for more meaningful interpretation of negative results. In quantitative methods, a high positive, low positive, and negative control should be included with each run. The high and low levels should be similar to critical points in the assay, such as the lowest detectable level of analyte. An acceptable range of variation for high and low positive controls may be established by repeated runs of each control sample on different days by different personnel. In PCR techniques, an amplification control is included as a target that should always amplify. The amplification control is used to distinguish true negative amplification results from false negatives due to amplification failure. Quantitative PCR methods that automatically analyze results require a standard curve or dilution series of a reference standard. In some instruments, standard curve must be run simultaneously with specimens, while others use previously determined curves that are loaded into the software. Alternatively, results may be calculated manually by linear regression of test results using standard curve data in spreadsheet software such as Excel. In methods requiring detection of a target-specific product or relative amounts of target, internal controls are run in the same reaction mix as the test specimen. For example, RNA from housekeeping genes is used for internal controls in methods quantifying infectious agents or detecting tumor cells by expression of tumor-specific transcripts. Centromere-specific probes serve as internal controls in FISH analysis, as do housekeeping gene probes on microarrays. In PCR, the internal control distinguishes false negative results from failed amplifications. The presence of an internal control also supplies a base for normalization of results. Failed normal controls in molecular testing are documented and require repeat testing of the assay. Quality assurance in the molecular lab requires periodic review and documentation of test results. Review might be, for example, in the form of numbers of positive and negative results compared with expected numbers from independent sources, such as published results over time. This type of monitoring reveals trends or shifts in rates of positive or negative results. Critical values that require physician notification are established by validation and confirmed by this type of monitoring. As with all quantitative testing, molecular quantitative methods need to have a defined dynamic range. The dynamic range is the ratio between the mean maximum signal of the target being detected and the mean minimum or background signal. This definition has been useful for describing the dynamic range of an assay. With quantitative molecular methods, sensitivity level and accuracy also need to be established and defined. The sensitivity level is when there's a change in the assay response with corresponding changes in analytes. And accuracy is the degree to which a measurement or an estimate based on measurements represents the true value of the attribute that's being measured. Assay levels that distinguish positive from negative results, also known as the cutoff values, must also be well defined and verified at regular intervals. Instrument maintenance is also an important component of quality assurance in the molecular lab. It must be monitored and maintained for consistent performance and accurate test results. Manufacturers supply recommendations for routine maintenance, and service contracts are used to provide support from professional service technicians. The laboratory maintains a schedule and instructions for all routine maintenance, such as checking temperatures, timers, and background levels. Parts are replaced as required or as specified by the instrument manufacturer, and maintenance schedules should reflect the amount of use of the instrument. In addition to routine maintenance and minor troubleshooting, such as replacing bulbs or batteries, repairs may be performed by the technologist with the aid of clear instructions from the manufacturer. These instructions must be readily available to the technologist in the event of an instrument malfunction, but technologists should also be aware of the limits of user-recommended repairs and when service calls are indicated. All the instrument maintenance, service calls, calibrations, and parts replacements must be documented. When reagents are replaced in a test method, ideally the new lot is tested on a previously positive and negative specimen as well as on the run controls. Instructions for the preparation of reagents and the quantities used in each assay are included in the written laboratory protocol for each procedure. Lot numbers and working stocks of probes and primers used in amplification methods are documented and matched to test performance in the runs in which they are used. The sequences of primers and probes are also documented, as any sequence errors made during ordering or synthesis of primers or probes will adversely affect test specificity or even results in the test, or they may result in test failure. Another component of reagents that are specifically involved in molecular testing are analyte-specific reagents, or ASRs. These are probes, primers, antibodies, and other test components that detect a specific target, such as a cell surface protein or DNA mutation. ASRs comprise the active part of home brew tests. They're usually purchased from an outside manufacturer and are classified as 1, 2, or 3. Most ASRs used in the molecular lab are Class 1. Several molecular tests are available as ASRs in infectious disease, tissue typing, and other areas of molecular diagnostics. Approved molecular methods include tests that utilize FISH, hybrid capture, PCR, and microarray technologies. Class 2 and Class 3 ASRs include those used by blood banks to screen for infectious diseases and those used in diagnosis of certain contagious diseases such as tuberculosis. Class 1 ASRs are not subject to special controls by the U.S. FDA. The test performance of Class 1 ASRs is established during validation of individual laboratory-developed tests. In the diagnostic laboratory, in vitro diagnostic, also known as IVD, reagent sets are intended for use in the diagnosis of specific disease or condition. IVD reagent sets include products used to collect, prepare, and examine specimens collected from patients. They should be used strictly according to the manufacturer's protocol. IVD reagents are classified according to performance risk and requirements for surveillance and verification, from Class 1 being with the lowest risk to Class 3, the highest risk. Regardless of the risk level, the performance, which includes validating the accuracy, precision, analytic sensitivity, reportable range, and reference range of in vitro diagnostic tests, must be verified by the laboratory in which they will be used. If an in vitro diagnostic protocol is modified, the procedure needs to be validated to show equivalent or improved performance compared to the original assay. A list of currently available FDA-approved tests is located on the Association for Molecular Pathology website. Chemical safety is also important in molecular diagnostics. The National Fire Protection Association has developed a series of warning labels for universal use on all chemical containers. The hazard labels have 3 parts, labeled with a number from 0 to 4, depending on the hazard level, in regards to flammability, health hazard, and instability of the chemical, with 0 being no hazard to 4 being severe hazard. The 4th section, Special Hazards, has 2 categories. O indicates a strong oxidizer, which greatly increases the risk of combustion. The stricken W, the W symbol with a line through it, indicates dangerous reactive with water, which would prohibit the use of water to extinguish a fire in the presence of the chemical. Most reagents used in current molecular diagnostic methods are not hazardous, and the increased use of automated systems reduces exposure to reagents. But still, some manual procedures can present chemical hazards. Volatile and flammable reagents need to be stored in properly vented and explosion-proof cabinets or refrigeration units. Secondary or reinforced containers are required for transport and handling of dangerous chemicals, such as concentrated acids and phenol. Radioactive chemicals are used in some molecular methods. Although methods involving radiation are being replaced by non-radioactive alternatives, Some lab procedures still use these agents. The Nuclear Regulatory Commission, the NRC, requires that laboratory personnel working with radioactive reagents maintain a radiation safety manual providing procedures for the safe handling of radioactive substances in both routine and emergency situations. OSHA, the Occupational Safety and Health Administration, has also developed regulations regarding ionizing and non-ionizing radiation. Radioactive reagents and methods are used in designated areas. Working surfaces are protected with absorbent paper, drip trays, or other protective containers. Potentially volatile radioactive materials are handled under a fume hood, and radioactive waste is discarded in appropriate containers separate from normal trash according to regulations. Test results in the molecular lab can come in the form of electropherograms, gel images, and autoradiograms, and should be sufficiently of high quality that the results can be determined unequivocal. The different molecular testing outputs should have clear bands or peaks without high background, cross-hybridization, distortions, or other artifacts. Controls should also be clear and consistent and reflect the expected size or level of the analyte. Molecular weight ladders on gels, autoradiograms, or electropherograms should cover the expected range of band or peak size produced from the specimen. For example, if primers used for PCR tests yield amplicons expected to range from 150 to 500 base pairs, the molecular weight ladder used must range from less than 150 base pairs to more than 500 base pairs. Documentation of assay conditions, reagent lot numbers, and quality and quantity of the isolated DNA or RNA is also required. In situ results, such as those associated with fish, are correlated with histological findings, so looking at the stained sections of tissue morphology. This is required when the molecular target detection is significant in specific cells. Documentation includes images of at least one normal cell with at least 2 abnormal cell results, and these images are cross-referenced or retained together with photographs, films, and autoradiographs generated from additional testing of the same sample. All the records need to be labeled with patient identification, sample numbers, target analyte, run identifiers, and the date of the assay. All of the raw data also needs to be retained with the final report and clinical interpretation of the test results. Careful documentation is extremely important because molecular diagnostic results may differ from results of other laboratories or from the clinical diagnosis. In general, when result reporting for molecular testing, they must be reported in terms that are unambiguous to readers who may not be familiar with molecular methods or terminology. The test report must include the method or manufactured kit used, the locus, mutation, or organism tested, the analytical interpretation of the raw data, and the clinical interpretation of the analytical result. This interpretation should include such interpretive complexities as the limit of detection of microorganisms, resistance factors, or the penetrance of inherited mutations, which is the probability of having a mutation but not getting the associated disease. The laboratory director, pathologist, or other clinical expert reviews the analytical interpretation, determines the clinical interpretation, and verifies the final results with an actual or electronic signature on the test report. When Class 1 ASRs are used in an analytical method, a disclaimer must be included in the test report. The disclaimer notes that the FDA has determined that such clearance or approval is not necessary, that the test is used for clinical purposes and should not be regarded as investigational or for research. It also states that the laboratory is certified under CLIA to perform high complexity clinical laboratory testing. This disclaimer is not required for tests using reagents that are sold together with other materials or instruments as a kit, nor for reagents sold with instructions for use. It's not necessary when the FDA-approved tests are used. In summary, important concepts regarding quality assurance in the molecular laboratory include proper specimen handling, which is required for accurate test results, proper specimen storage conditions so nucleic acids are preserved, molecular test performance being monitored by the use of quality controls, instrument maintenance and calibration for accurate detection and measurement of analytes, proper reagent preparation, storage, and use as recommended by the manufacturers and/or laboratory protocol, and finally, appropriate documentation and resulting of final results, including saving raw data and clearly reported test results.